So first of all, thank you, Kushalaida, for giving me the opportunity to present this work at this machine learning reading group. I'm audible.
Yeah, you're audible.
Yeah. So as Kushalaida mentioned that I am a second year PhD student at Indian Association for the Validation of Science, Kolkata, Jadavpur. So basically, my work mainly focuses on application of graph neural networks and NLP on um, drug target binding affinity predictions or material science property predictions or materials property predictions or some some properties predictions on proteins as well as genes. So so here I will give a presentation on my work on our work DeepGLSTM. So deep graph convolutional network and LSTM based approach for predicting drug target binding affinity. This work is accepted at SDM 2022, and the co-authors of these works are Madhusudan Ghosh and Dr. Partha Chaudhary. So at first, I will give the basic introduction. What is graph target binding affinity prediction? So first of all, I will before dive deep into the graph target binding affinity prediction, I will give a brief overview. What is drug repurposing? So we have all came across the current pandemic situation like COVID-19. So at this pandemic situation, we rapidly need to generate new drugs or the drugs which are already approved by the Food and Drug Administration of USA on other diseases like lungs cancer, prostate cancer, breast cancer, or Ebola. And can we apply those drugs on newly come disease like COVID-19? So this is our main uh, this is our main problem definition. This is our main motivation that why we apply AI in drug target binding or even apply AI in drug repurposing. Now, drug design is basically defining um, divided into two parts. One is generating new drugs from the existing drugs, or another is repurposing the drugs which are already available in the market. So, in the earlier days, drug design is very hard because we from the literature we know that the number of druggable candidates present in the nature is something around greater than 10 to the power 8. So it's very huge. So if we without applying those, the main pipeline follows is first we take those large candidates, and the chemistry person, that is the experimental chemist, take those drugs, and from those drugs he or she will get the possible candidates that can be applied to on a particular protein. But and after that the clinical trials are happens, and finally the drug is administered by the FDA that can be applied on a particular protein on a particular disease. So but the first stage, that is the using the candidate drugs using a wet lab experiments, is used it takes a it's a very time consuming process, because at this stage the wet lab experiment is very hard to process, and also there are some external dependencies on that. So for this reason, AI helps for to overcome this problem. AI comes into the play, and using AI, we can generate those. We can get the possible candidate drugs without applying the wet lab experiments. And after getting those possible candidate drugs, we can take 10 top 100 or top 10 top 100 of that to in the wet lab experiments, and from after that we can conduct the clinical trials. So, for example, in the Spanish flu pandemic, the generation of new drugs takes 10 to 10 to 15 years, which is very huge. So, if we depend on that, then we can overcome the COVID-19 problems. So, this is the main motivation that why we have applied, why AI have came in the drug repurposing or discovery problem. So here we focus on drug repurposing problem. Now, drug repurposing problem is typically deals with drug target binding affinity predictions. So now, what is drug target binding affinity prediction? Now, drug target binding affinity predictions is a problem where we take the drug molecules and their corresponding target protein and find whether this drug molecule can bind with this target protein or not. Basically, it produces a score, and on the basis of score, we take that drug as the possible candidate to proceed the further experiments, that is the another wet lab experiments or the clinical trials. So this is the main problem definition. This is the problem definition of drug target binding affinity prediction. From this figure, we can see that this is the large scale data sets. Typically, in this domain, Davis KIBA are used. I will discuss about the datasets in the upcoming slides. So this is the drug molecules and this is the target protein, and we can apply AI in this part, that is the deep learning or machine learning or any kind of AI models, like the graph neural network based architectures or the things to encode those things, and from that we will infer whether this drug is a possible candidate or not. So this is the main problem definition of our work. So here we see that the drug is bind with its corresponding protein at this position, and from that, if it produces some score greater than some threshold, then we can proceed this drug as a candidate drug against this target protein, and we can proceed the further wet lab experiments or other clinical trials, and if not, then we can exclude that drug. So now, why the importance of this model on current scenario? But basically, in the work that I have done in the COVID-19 situation, so that time we have to generate new drugs or to repurpose the old drugs against SARS-CoV-2. So this is the main motivation that we obtained. So basically, if in this figure, this person has some symptoms of COVID-19, the typical COVID-19 symptoms like fever, sore throat, headache, running nose, shortness of breath. So he came to, or he or she came to doctor, and doctor will prescribe him to test the RT-PCR, so which is a commonly testing technique that we use against test whether the person is affected with COVID or not. So after getting the he inferred as the positive, so he came back to doctor, and at that part, if doctor have to design a new drug, then it will take 15 to 10 years. 15 to 10 years by using, if we apply those traditional wet lab techniques. Now, at this position, if we apply some AI-based techniques to screen those drugs that are already been used for other diseases and apply, and from that, if we choose the possible candidates and then we apply the wet lab experiments or other things, then the doctors to prescribe drug is very easy. So for this reason here, the main motivation that we have applied this architecture or this work against SARS-CoV-2. So, or SARS-CoV-2 or any kind of newly came diseases. So I will give the main architecture of our work. So this is the complete architecture of our work named DeepGLSTM. So basically, it has two blocks: one to encode the drugs and another to encode the protein sequence. So here, drugs are in computational chemistry. Drugs are typically denoted as the SMILE strings. So SMILE strings are the strings where a molecule is encoded to that if molecule is encoded to apply computational methods on that. That is, it's a machine readable format. So now here we see that carbon is C is denoted as the carbon, equal to sign is denoted as the double bond, and this is the 1D representation of the SMILE string. So you can apply any kind of, there are several kind of softwares, chemotherapy softwares to convert the SMILE string to corresponding molecular graphs. So here we apply the widely available, widely used chemoinformatics software called RTKit to encode graph SMILEs to its corresponding ambiguous representations. So and then I will discuss each block in my upcoming slides. So basically here we take these the representation into feeding in these three blocks, and after getting the we predict the output. Now basically, since here we predict some affinity score, and this is a continuous value, so this is basically a regression problem where we train this model using a mean square error loss function, and which will be updated the whole the model parameters. So now I will give the main input block of our work. So now drug SMILEs, in the previous slides, I have said that drug SMILEs are typically encoded as a 1D string structures. So if we apply, there are some existing library called RTKit. Then if we apply RTKit on top of the smile strings, we will get this as a representation. So now, what thing that we have done here is basically what the GCN does is it takes the source nodes embedding and updates its embedding by getting the neighborhood nodes embedding informations. So this is the way the GCN works. So since this is the main motivation that we this is the main motivation. So why we applied a square a cube in our first block, that is the input block of the drug encoder. So here we basically take the A, that is the original against representation of the drug smiles, and then we apply a square, that is we apply the power curve representation of A, that is the multiplication of A with A, and for the last block we apply A square with another multiplication with A, and then we get A cube. So why we applied these things, I will discuss the next slide. So basically, this is the input block. This is the how we format our input representation of the drug smiles into the GCN encoder. So this is the main GCN block descriptions. So I have previously said that GCN, what the GCN does is it takes the source nodes embedding and generate its and updates its embedding by getting the 1 hop, 2 hop, or 3 hop neighbors embedding. And here if we apply A on top of this first layer of the GCN, then it will compute some kind of A square thing. That is also the power representation of A. That is the A square also captures the 2 hop neighbor information in it if we do not use the GCN layer on A. So here that is the first block of the first layer of the GCN will simulate the A square things. Similarly, on top of that, if we apply another layer of GCN, then it will simulate the A cube. If we do not apply GCN on A, and if we extensively apply, if you extensively multiply A 3 times. So another on top of the GCN layer, if we introduce another GCN layer, then it will compute the A to the power 4. So our main motivation is to simulate A with GCN and simulate A square with 2 GCN and simulate the main simulate the whole things that is computed by this block. If we inputted A square in this block, that is if 2 GCN is paired, basically here we see that in this block of GCN we apply A square on a first layer of GCN. Now I previously said that the first layer of GCN captures the A square representation. If we extensively apply A square externally and feed into the GCN, then it will compute A cube intuitively. And similarly, if we apply GCN on top of that GCN, then it will simulate this block, that is it will compute it to the 4. And similarly here, so the number of GCN layers reduces by 1. Similarly, for this GCN block, if we apply A cube on that, if we apply A cube on a single GCN layer, then it will compute it to the 4. So this is the main motivation. This is the intuitive motivation that why we have applied A squared A cube in the GCN encoder. But in the ablation start, we have also performed the ablation experiments that why we have applied only 3 layers of GCN. So intuitively, the main reason of applying 3 layers of GCN is the number of nodes present in a small molecule is not so much large. It's not so much big because the small molecules are the size of the small molecule graphs are typically small. So also we studied our overall data sets and get to know that the shortest minimum, the number of shortest path lengths present in the molecular graph is not much more than 3. So if we apply GCN layer, 4 GCN layer or 5 GCN layer, it will generate the source nodes embedding repeated time, same time. That is, if we apply 3 layers of GCN, what happens if we apply another layer of GCN, then it will the computation was more increases, but the results is not so much good. So for this, the main motivation that we applied only three layer of GCN on that. And also then the previous studies they also applied 3 layer of GCN. This is the main reason. So after getting the representation of this is the first block's representation. Second block of this if we apply a square and then we get 2 GCN layer on that, then we get this representation. And if we apply a cube and on this GCN layer, we will get this representation. So this is the GCN block part of our architecture DeepGLSTM. So does have any question towards this point, or I will.
Hello.
Uh,
I have a question. I heard like you tried to explain that why this A square is equivalent of having 2 hop information, but somehow I was not able to get it. So can you please explain once more?
Now what the GCN does is GCN basically takes the nodes information and it source node information and learns its information by getting the neighborhood nodes information. Now if we apply GCN on top of A, that is the original adjacency matrix, it intuitively computes A square. In A square, we also have some sort of 2 hop neighbor information if you multiply A with A. And also
And that's
GCN
what I am
intuitively.
a bit doubtful about. Like, does A square, like A is the adjacency matrix, so I multiply with it itself. So I'm not able to get the picture. Like, how does it calculates the value about like information about or knowledge about the adjacent
Yeah,
neighbor? Or
basically it A what the A square does is it, in the diagonal if there is no self loop present in the node, in the diagonal they are all 0, and if we apply A square, then the self loop is already explicitly included in the, it becomes a weighted graph. So in the weighted graph, some sort of 2 hop neighbors information captures. If you do this by hand, you can get some of 2 hop neighbor information, but this nodes are non-neighbor. So to get the non-neighbor representation, we apply GCN on top of that. And from the previous literature, our representation captures some sort of 2 hop representation, but not in so it is a weighted, not in a non-level features. So for that, we apply GCN on that to get those learnable features. Basically, we take the features also, that is the node features, and apply the node features on the 2 hop representation of A, that is the A square, and then we propagate GCN on that.
okay, and the output of all these three these blocks which you have mentioned, like 3 GCN on A, 2 GCN A square, and single GCN on A cube, the equivalent, or I will say the logical output of all the three is same, that is A power 4, if I'm not wrong.
Yes.
Then
Right.
why did we have this 3 different, like why can't only single A4 representation?
Since here we applied this because in the molecular graph we have to capture more the subgraph representation, because in the molecular graph there are some subgraphs called the functional groups. If we did not get those information in a quite well, then the molecule will not, then the GCN representation will not act as the molecular graph original molecular representations. In the upcoming slides I will give the explainable part why we have applied, why the square representation captures the functional groups or other structures in the molecular graph. So we need those to capture those structures, like the functional groups, motifs present in the molecular graphs. So for this reason, we have applied we get basically to the weighted graph representation. That is, we take the same graph and change its orientation and get another weight on that. Then how they apply, how the increase the model's performance. So in that part, we can apply in the upcoming work. I have applied some mathematical notations, why mathematical theory that why it happens in the inside the GCN if we apply a square, a cube, or other things. The first part. This is the first we have applied this in the molecular representation to capture important motifs.
Okay, Sriman, just a small question. Actually, I have 2 questions. One is that as you are saying, like once it is passed through 3 GCN layers and it returns you a power 4. So I mean, so is it exactly the same as a power 4? Like
No,
if I multiply it a 4 times, whether it will give the same thing? And my second question is that it's actually replicating four hop counts, right?
yes,
So it
yes.
is capturing 4 hop counts. So is there any limitations of a hop count, or it is like the problem specific thing? That's why it's implemented in 4 hop count.
Yes, this is a problem specific because I previously said that the minimum in the datasets we get that the minimum photos path length is not more than 3. And also in the also I have said that molecular graph is not so much big like other graphs. So if we since this is not a polymer, if we consider as a if we consider polymer as the molecular graph, then the number of nodes will be increases. But here we consider the small molecules because the drugs are basically the small molecules. Like the number of nodes is not much greater than uh, like 20 to 30. I have to you can get the statistics from the GitHub also if you parse the number of nodes, how many nodes the datasets have. So for this reason we have applied. Also in the evaluations part we also came to know that if we increase the GCN in the number of layers of the GCN greater than 4, the performance of the model decreases. And your the first question is I cannot the actual if we apply GCN on 4 GCN on a similar is equal to the AQ1 GCN layer is the first.
Yeah.
So in this is in the first question that I have understood that it is kind of similar but not so much main because it's not so much the embedding will be not same. There are some changes. That is the weighted representation.
Okay, understood.
And I think, yeah, in every layer of GCN, you do some sort of non-linearity also. So that's why every time this A will not be exactly multiplied to there. So there are some non-linear part in between will be there. So that's why if you apply 3 GCN, yeah, if you apply 3 GCN, you won't get exactly A to the power 4.
But
so
some sort of
we can imagine that, right? Intuitively, we can say that this is 4 times getting multiplied. So intuitively, we are saying it.
Yes. So one layer of GCN means you are collecting information from one hop. So if you're applying 3 times, so intuitively you're collecting from 3 or 4 hops. I mean, whatever you can think of. So that is fine, but exactly it won't be like this because every hop there will be some transformation also happening.
Right.
So
right.
What you have said that you can. So okay, so basically these the thing you have said that it will not same, but some that if we get the cosine similarity of those two embedding, it will be higher.
Yeah, it will be similar. Yeah, intuitively it's
similar, kind of 90% or greater than 95% kind of percentage of.
And regarding that particular question, I think Chetan has asked that why A to the power k is kind of k hop. So I can't remember exactly the theorem. So there is a theorem that A to the power k actually signifies number of k length paths between two nodes. So that means that how many ways you can collect message from two nodes. A to the power k means. So this is how actually it's similar to kind of you are applying k layers of GCNs. Intuitively, you can think like this. So what actually Simon is trying to say.
This is the main intuitive implementation. Intuitive.
I'm trying to find the formula theorem. If I find out, I will paste.
Okay. So now this is the. So now can I proceed? Is there any other questions? So I think.
Yeah, you can proceed.
Yeah. So now this is our second block of our architecture is called the BioLSTM block. Here, this is simply it applies the BioLSTM, the bidirectional LSTM model. Everyone know about this the architecture. So basically, the protein sequences are denoted as the amino acids. Here, M is an amino acid, K is an amino acid, F is an amino acid, and R is an amino acid. And so, if we apply, so basically here we take the embedding layer, that is we map with the English alphabets present in the dictionary, and we present in the English alphabet dictionary, and we get the one-word representation of the protein sequence. So after that, we will apply a BioLSTM layer. Now here, why we apply BioLSTM? Because the length of the protein sequence is 1,000. Since it's very weak, to capture the long range dependencies, we need the BioLSTM layer. So nowadays, this work is from 2022. Nowadays, there are some ESM transformer models are also implemented on proteins to capture the information, and also we can convert the protein sequence into the protein graph. That are also doing that things. But to generate the protein graph, the main limitation is since the protein structure is very big. So if we take those big graph and train the model, then it will become very much cumbersome. So for this reason, here we take the protein sequence as the LSTM block, LSTM to capture the protein information from the protein sequence. So after getting these two representations, that is the three representation from the graph convolutional network blocks and the protein representation from the LSTM block, we concatenate these three things and apply global max pooling, and apply some linear transformation on that, and for the protein representation we also apply a linear transformation, and then basically we concatenate those two things and we apply also to fully linear transformation to get a single output. And since I previously said that this is a linear regression, so this is a regression problem, so we apply mean square error loss to update the model's weights. So now these are the results. So there are some existing datasets called Davis, KIBA, DTC, and also in the next slide there are other three datasets. So now what is KD? Now in the biology literature, the affinity values are typically denoted as kinase dissociation constant, kinase inhibition constant, half maximal IC50, AC50 kind of things. So here KD is a affinity score. So now what is PKD? Basically the KD value is so much big that we have to normalize it. We need to some lower value. So for that we applied some minus log 10 on KD, and those things is denoted as PKD. So here Davis, since Davis is a KD value, so we apply PKD on that, normalizes. KIBA is another dataset which is generated by taking all the affinity scores and they normalize it into a score, and they call this as KIBA. And how they normalize, you have to go through their paper, of the paper that first present the dataset KIBA. And from DTC there is another metric called PKI, where KI is kinase inhibition constant, and if we apply log on that, we will get the PKI. So here we take the existing SOTA, that is a GCN, GAT, GIN, and if we apply, and another is the DeepGLSTM. From this figure we see that our models performs well on Davis datasets, also KIBA, and also DTC. That is the full form of DTC, drug target commons. And for the Metz, and this is another experiments on the other three datasets, which is Metz, ToxCast, and the STITCH. Here the Metz has also the same affinity metric as the DTC, which is PKI. And for ToxCast, here the affinity metric is AC50. And for the STITCH, the affinity matrix is scores. That is the score that is present in the STITCH datasets. So basically here our model performs well on these three datasets. Now here we measure our model's performance against these three metrics: mean square error. CI is the correspondence which is the traditionally applied in the regression problem. That is in the regression problem. And RM square is another metric that is typically used as a statistical measure in the regression problem. And since this type of techniques are typically known as QASR. In QASR kind of models, RM square is a very good metric. Here, if the mean square error is less, then our model is good. But for CI and RM square, if the value is greater, then our model is performing well. So these are the results. So after that, we have.
Yeah. So I have a question here, Simon. So what is the size of these datasets?
Typically, the Davis datasets has 66,000 drug target pairs. Basically, it consists of drug and corresponding target. Target means the protein sequence, and there is an annotation value which is annotated as KD. That is the kinase dissociation constant. And if we apply log on that, then we get the PKD. Similarly, I think KIBA has the number of datasets and the number of samples present in the KIBA is greater than 1 lakh. This is the largest number of samples present in this. For this reason, the model performs well in KIBA because it gets the higher number of training examples.
Okay.
But DDC because
And
it's
one more thing.
Hmm.
Yeah, and one more thing. This
Uh-huh.
SOTA model you are saying GCN, GAT, and G. So these are all graph
Yeah,
models, right? So
Uh,
yeah. So these are all graph models. I feel these you have used for the molecule part. But what you have used for these protein sequence part there?
Basically, these things are the existing work that have done before this work. Like the graph DTA, there is another paper named graph DTA. In that part, they take the 1D CNN to encode the protein because this is a very old paper. And they basically first introduced graph convolution neural network in this task. Before that, only the string-based of techniques are used to encode the smiles. Because smiles is a string, we can apply any kind of NLP techniques to encode that.
Yeah, that I understand. I'm saying graph part I understand. I'm saying that there is another part that is protein sequence, right? How these SOTA models are processing the protein, which you used LSTM for. I mean, you have used LSTM for processing the protein
SOTA model,
sequence. So how these
so for SOTA model, they take the graph DTA take 1D CNN, 1D convolution unit.
Okay. Got it.
I have a question. Hello.
But
Yeah.
seen.
Yeah. So my am I audible? There's a voice break.
Yeah, Sriman, your voice is breaking. I
Cannot hear.
Yeah, Sriman, your voice is breaking. Hello.
Hello.
Yeah, now
In
it's fine. Yeah.
the main reason why we have applied, I have also did not forgot to mention that the main reason that we have applied graph convolution is to capture the topological information. Because if we apply neural NLP-based techniques or smiles, then it cannot capture that we applied that the nowadays graph neural network have been applied on molecules. So I have forgot to mention that thing. You have asked the question and I remember that.
Okay. So Chetan has another question. Yeah, Chetan, you can ask.
Yeah, so I would like to discuss, understand more about the problem. So the data sets are in such a way that there is a drug structure and a
Your
protein,
call is
and the
still dropping.
Am I audible now?
Yeah, you're audible. At least I can hear you. Simon, you can hear us?
Yes. I am audible.
Yeah, so I just want to discuss, understand more about the problem. So the thing is, these data sets have a protein structure and a drug molecule and some value associated with that couple. So should I type
Not protein
it?
structure.
Yeah, probably you should type. He can see.
Okay.
Maybe at the end we can take it.
Not
Simon, you just complete
okay.
and then maybe yeah.
So this is the results that we have obtained if we apply our model on these datasets. So now this is the case studies that we have applied on SARS-CoV-2 viral proteins. Now basically we take known protein structure that is present in the SARS-CoV-2 like spike protein, helicase, papain-like protein, etc. So now we go to the the person who knows biology better than me, and then say that there are two types of drugs present in the literature. That is the drugs which bind with the receptors, the drugs that doesn't bind with the receptors. Now, what is receptor? I did not know. I know about very much things about that. They say that. So, and for getting the information from them, I basically curated some drugs which are already approved by the Food and Drug Administration (FDA) from drug bank or drug bank datasets, and sort those and divide those two drugs, the collection of drugs, into the drugs which are bind with the receptor and the drugs which are not binding with the receptors. And then we apply our model on that. So here we also apply pKi, but in our main paper we only apply our model. We apply the model which is trained on pKd and K. Now, since these two data sets are widely used in this domain, we can get from the literature that everybody have applied this applied their model on these two data sets, which is Davis and KIBA. But in our work, I have additionally curated on more data sets like Metz, ToxCast, etc. So since from the literature we previously know that there are only widely used data sets are KIBA and Davis. So for this reason, we only take the pKd value and the KIBA score to measure the affinity prediction in the case studies. And from that, if we to combine these two scores, we basically take the average of that and we take those scores. If this score is high, then our model is, then we can take these drugs in the wet lab experiments, and if it's less, then we can reject it. And here from the biological people, we first I short those whole FDA-approved drug into two subsets, and we apply on our model on that, and we came to know that this drug has the highest affinity score. And from previous literature, we also that some of these drugs have already applied on SARS-CoV-2. So since this work is from nowadays, there are various kind of new drugs, but we have over the time when I work on this project, I get these are the top possible drugs can be applied against SARS-CoV-2. Also, we have applied some wet lab experiments in our biology lab, so we get that they also said that this can be applied against SARS-CoV-2, but we have to perform the clinical trials also. So these drugs are already available in the market. The biological person also buy this drug from the their vendor, and they also apply in their wet lab experiment. They basically take the spike protein and heat the drug on that, and they get some results. And they say that it can be applied in SARS-CoV. To conduct this, we need the the clinical trials. So similarly, we also take another two proteins, which is RNA dependent RNA polymerase protein and the WRF3A protein. These are also present in the this also present in the COVID-19 in the SARS-CoV-2 virus. So now this is the main. Now after that we will get this is the main part of our work, that is how our model captures the important functional group present in the molecular graph or not. So now here we see that from the literature we came to know that if this kind of subgraphs present in the molecule, then the molecule can be toxic or that there is some structural alert. So now there is another chance that the drugs which have our model predicts that it can be applied against the SARS-CoV-2, but it can have some structure, it can have some side effects like toxic side effects. So to remove that, we have also applied some sort of explainable things, that is we apply some color map, that is at a higher the color means our model gives higher weightage on that node, the lower means the weightage is less. So here see that the dark blue means the weightage is less, and the dark orange means the weightage is high. And from that we see that this is the baseline model which is proposed by Graph Details, the graph attention network, and this is our architecture DeepGLSTM. We see that in this part our model gives higher weightage, but the Graph Details gives lower weightage. So from that we can discard this drug, that it can have some structure alert present in this. So basically this is the main crux of our work, that why we have applied 3-layer of the power grid representation. Basically it captures some important functional groups present in the molecular graph. Similarly, in this figure we see that this is another molecular, and this is another drug, that if these two structural group present in this molecule, then it can have structural alert. But our model gives higher weightage on that. Basically it captures higher information in this part, but the baseline model fails to do that. So now I will give the ablation study of our work. So previously I have discussed that why we have replaced 1D CNN with BiLSTM. So this is the reason that first we take the 1D CNN and the result is this. But if we apply BiLSTM layer, because the length of the protein is 100,000 and one DCNN fails to capture the long range dependencies from the literature. We all know that, but the BioLSTM captures that very much well. So for this reason, it performs good. And similarly, this is the another application study that if we take the first layer of GCN, if we take the first block of GCN and we feed A on in that, what it does, and if we take two layer of GCN and apply A square on that, what the results produces, and if we apply our DeepGLSTM, and finally if we increase the number of layer with 3 to 4, then what it happens in the model performance. These two basically same things but represent in a different manner. That is the effectiveness of power graph and the effectiveness of why we applied different components in the DeepGLSTM. So now, so this is the final concluded part of my presentation is basically in this work we apply FDA-approved drug to predict the affinity values between the target proteins and the FDA-approved drugs. And from the results part, we know that our model presents state of that results at those situations. But in the current scenario, there are very much paper that they produce more state of the results. Also, our model use 3 blocks of GCNs, that is the first, second block, and captures the power graph representations to learn the topological information of drug molecules very much well. Also, our model has another block, DeepGLST by LSTM layer, which captures the protein sequence information. And finally, we take the FDA-approved drugs and we perform how much will how and we get the binding affinity values between those FDA-approved drugs and the target protein, and infer which drugs can be applied against SARS-CoV-2. So this is the references, and this is the team that worked. Me, Munsunuko and Parthu Choudhury. This is the archive version of the paper that you can get from this QR code, and this is the GitHub link that you can go through the link, and there you can run the model. So thank you.
Yeah, so any questions?
Yeah, so as I was saying, I was about to ask, I want to understand more about the problem statement. So tell me, let me know if I'm wrong. So what my understanding is, these data sets, each instance of the dataset is like this, like there is a protein structure and the drug associated and number related to this couple, like how much this drug is effective in binding with this protein. So like, is there any limit or like any range of this number, and what does this number really mean?
This number is basically the data sets are basically formed in the wet lab experiments. In the wet lab experiment, they measure those values by taking the original protein structure and the original graph, original drug, and they bind in the lab. The wet lab experiment mean in the lab that is present in the chemistry person that they have done the experimental chemist. So from that they basically get those values. So for the affinity prediction, there are mainly kinase division constant is used, that is KD, and there are other data sets that I mentioned. There is Metz, ToxCast. They basically used ICKI, that is kinase inhibition constant, IC50, AC50 kind of bioactivity values. Basically, these things are called bioactivity values. They're basically generated by the biological persons, not.
Okay, so
The
this
computational
number
biologists
is
were.
not standard. This differs as per the dataset.
This KI is not same as the KD value. For this reason, we have to apply our model on various kind of data sets.
Okay, and can you please go back to your result slides?
This.
Yeah.
Like,
so yeah, this one. So the thing is, like, I'm not from this domain, so I'm just wanted to ask, like, how much improvement do you say that is a good improvement? Because in one of the pictures, I can see that your proposed method is below the performance of the SOTA. In other majority it's somewhat like nearby, like maybe 1 or 2% more. So is that considered as a significant improvement, or like I'm just asking, I'm not from this domain, so
Really, I have previously mentioned that the number of datasets present in the device datasets is so much small, like 66,000. So if we apply train test splits and train our model, then our model will not get so much information, because to train GCNs or LSTM, we need a large number of data. And the time that I have applied this work, I have done this work, there are the application of transfer learnings or any kind of things not done in this domain. So we can apply those things and we can increase our model's performance. Some of the paper that I've cited by this, that also cite this work, they have obtained better result than our work. But at the point that the time that we have applied, we are the second one to apply graph convolution network on this field. So for this reason, that you are basically did not get not so much, did not focus, not so much get the good results. They say that they are the results are impressive, because for one data set it's not so much good, but the other data set like KIBA DTC results are significantly better. And similarly for this also. And we are the first one that we apply our architecture on all of these data sets. Similarly, typically in this domain, the application of models is only used, the models are only trained by Davis and KIBA, but we have applied it on other datasets also. So there is some small improvement, but this is a very much big, because we need good models. And nowadays there are very much good models. They have also improved our work also.
and if we want to compare the size of these models, then can you please comment on them?
Size means not so much big, because GCN, I did not remember that the size of the model, but
No, if
not
I'm
so
thinking
much.
like if SOTA is a big model and your model is smaller, then even if you have
No,
marginal improvement, that will be considered better because your model is smaller than the SOTA.
and there are SOTA models have also not so much. I did not remember the model size, because
So like, is it comparative, like similar size?
kind of our model kind of similar size, because they also apply GCN, but our model has some more parameter because we apply LSTM on that. Some the other
Okay.
gates are also we preserved. But nowadays in this domain, LSTM is not used. They basically use BART models, that is the BART, Rosetta BART, or ESM, the Facebook model, that the ESM model. We can get the ESM embedding from the proteins, and we can apply those embeddings to encode the protein structures. That will produce good results. At that time, around 2022, the application of ESM based models on these domains is not so much widely used. But so for this reason, I have applied LSTM and we get the results. But if we apply Rosetta BART or the ESM model that published by Facebook, they will improve the results, but much more better than these.
Okay. Now,
Actually, the work that I've
Mm-hmm.
These are.
Oh, yeah, please continue. You were saying something.
No.
Okay, then we can go to the case study slide. Yeah, so like I think I missed you. Like, what do you want to cut away from this? Because I was like in the previous slide, you were showing the comparison between SOTA and your model, but here I can't. I'm not able to understand. Like, so can you please explain these figures?
Now, in this section, we basically apply our model in the real world situation. That is, there are some protein present in the COVID-19, like spike protein is a widely, this is the high protein that have present in COVID. So if we take those in the database known as UniProt, that from that we can get the string representation of the spike protein. And there are some databases like Drug Bank, which has some FDA-approved drugs. The drug that is already approved by FDA for other diseases like cancers or Ebola, some sort of things. So if we take those drugs and apply our model, that is, we basically take those the pair, that is the spike protein and the corresponding FDA-approved drugs, which is not used in COVID-19 but used in other diseases. And we apply our model on that, our model will produce some value like KD, that is the model which is trained on Davis dataset, will produce the kinase dissociation concept, PKD. The model which is trained on KIBA, it will produce the KIBA score. So these things have done here. Basically, this is the prediction time. We are not the performance. We will not compare the performance of the existing model with this, because they did not apply on that, because we applied these things in the COVID-19 situation, and the work that have done before this work, they are all not in the COVID-19 situation. For this reason, they did not introduce not so much case studies. So this is the reason we have applied. This is basically the how our model predict the affinity values.
Okay.
Okay, anything else? Anyone have any other question? Okay, then let's thank Sriman. Thanks for this wonderful talk. And we'll wrap up today's session here. And again, we'll meet in the next session in the coming Friday. Okay, thank you all for joining.
Thank you all. So